So I'll get started with some, as more people join us, I'll just get started with some initial housekeeping for the webinar session today. So again, just thank you all for taking the time to meet with us today and learn about ERGA. We are really excited to share the European Reference Genome Atlas project with you, and also the BGE project with you, which you'll hear more about, and the BioSCAN project with you as well. So a lot to cover today, and we hope that we will keep it within 1 hour to be respectful of everybody's time. If you do want to contact us, I will be sharing the slide deck to this with you all, so no worries there. And all of our contact information, our email, our website, and our Twitter are available on the slide deck here, so you'll have that at when I share the slide deck. Just some initial housekeeping. At the end of the session, I will be doing a poll. This is an anonymous poll and only for national focal points. And the reason for this is that there's a cap on the number of people that can participate in the poll, and that's around 30 participants. So if only NFPs could register through this link, and I will put that link into the chat for people just to click onto. Do that now. That should be available to you. So if you just click into that, you should be able to participate anonymously in the poll, and that will be coming at the end of the session. So I've already gone through. If you could rename yourself, that would be wonderful, with your first name, the country that you're representing today, and just indicate that with ended with a national focal point in brackets. That would be very helpful for us. If you have any questions that you would like to present to any of our speakers, our panelists today, please feel free to use the Q&A function to share your questions with us, and we will floor those questions at the end of the presentations of all of the panelists. If you would like to share your question verbally at the end in the open dialogue, please feel free to raise your hand. That way we can mute you and you can raise your question verbally if that's your preference. If you have any resources that you would like to share with us today, please feel free to share them with us through the chat box function. That would be wonderful. Yeah, just for your own reference, this session will be recorded, and we can circulate all of the recording, both the recording and the slide deck after the meeting. So thank you again for coming, and to set us off today, I would like to introduce Claudio Cioffi, who is from the University of Florence, and he is the Vice Chair of the European Reference Genome Atlas, and he's going to set us off with a 7-minute presentation about giving you an introduction to what is the European Reference Genome Atlas. So Claudio, I'll pass it over to you, and you let me know when to proceed with the slides.
Thank you, Anne. I'll first like to briefly mention the European Commission's priorities, the first of which is the European Green Deal, as most of you are aware of. And there is a section in the European Green Deal specifically addresses preservation and restoration of ecosystems, biodiversity, and genetic resources as crucial priorities. And basically, our initiative is, let's say, embedded in the Green Deal, as it provides background knowledge on the genetics of European living organisms. And it is actually the first of its kind to do that in a concerted fashion through a network of institutions across the whole European Union and beyond. Next. So the initiative is called European Reference Genome Atlas, and is a pan-European consortium involving nearly 50 among EU countries and other partners, with the aim of generating reference genomes. So reference genome is basically a high quality, nearly error-free DNA sequence representing the entire genomic makeup of a species. So generating reference genome representing basically the European biodiversity, and these ranging from endangered and threatened species species that are important to ecosystem function and stability, and key species for agriculture and fisheries. So in the next slide, you can see the next. Okay, the ERGA started basically at the end of 2019, was conceived in early 2020, and basically the number of people grew during 2020, and 2020 we basically sent a position paper to the EU Commission advocating, say, the importance of genomics for biodiversity conservation. And we were able to give our contribution to redraft the first call of the Cluster 6 panel of the Horizon Europe project that was dealing specifically with the importance of genomics for biodiversity monitoring and conservation. Next, then in 2021, basically we had our council established. The chairs were elected. Different committees that that are included in ERGA were formed, and we start having a number of activities, including, for instance, the community call for species nomination. So basically the members of ERGA can submit, say, a number of wish list of species to be sequenced within the ERGA framework. We had a forum article published in Trends in Ecology and Evolution, and then there were two main activities that were started: a pilot project and a joint ERGA BioSCAN Horizon Europe project, which Rob Waterhouse will talk a bit more later on. So next. So now we are basically more than 700 members across Europe. Next. And the goals are basically the main goals are basically to establish a pan-European genomic network and connect the existing European infrastructure for establishing a high-quality genomes. These are short-term, mid-term goals, and basically the mid-term and long-term goals are to establish reference genomes of the entire European eukaryotic biodiversity. Next. So apart from let's call it executive board, so it's comprised of the chairs, one chair and two vice chairs. ERGA has scientific committees and the one that we call Science Plus committees. The scientific committees are comprised of members of the ERGA community that are interested in sample collection and processing, DNA sequencing and assembly, annotation, data analysis, IT and informatics, while the Science Plus committees deals more with ethical, legal, and social issues, media communication, citizen science and outreach, and TKT, training and knowledge transfer across the ERGA communities. So next, so basically ERGA is it's really a kind of multidisciplinary, let's say, initiative. Here you can see that from sampling of collection of samples to taxonomic identification, DNA processing, sequencing, assembling of genomes, and data analysis. We basically include different types of institutions, from the universities, of course, museums, specialized molecular labs, sequencing center, and bioinformatics and computational groups. Next. So, as I was saying before, one of the first, say, initiative of ERGA has been and is currently the pilot project. Basically, pilot project. The aim of the pilot project is basically to produce genomes for a number of species proposed by the ERGA community, and then in particular to test, say, the distributed model of ERGA, that is the joining of different sequencing facilities and different types of expertise across Europe, with a common goal, of course, of producing reference genomes of the European biodiversity. And the second large initiative is the joint venture next with the, okay, this is a slide that probably Anne will show later on. Anyway, this is just an example of the number of species that we are working on, that the pilot project is working on, and you can see kind of the diversity of organisms that are included in the pilot project. And then the second large initiative is, of course, a European Horizon project, which is called Biodiversity Genomics Europe, which is basically a joint venture between ERGA and another initiative called BioSCAN Europe, which deals more with single, say, section of the DNA for barcoding species, so for recognizing single species. And I think I'll hand it over to Rob, which will talk more about the Biodiversity Genomics Europe project.
Yeah, thank you so much, Claudio. So next on our panel list, we have Dr. Rob Waterhouse of University of Lausanne in Switzerland, and he is also a vice chair of the European Reference Genome Atlas. And he's going to give us an introduction to the Biodiversity Genomics Europe project. So I'll hand it over to you now, Rob, and just let me know when to change the slides, and you have about 7 minutes as well.
Thanks so much, Anne. Yep, so this is really, like Claudio said, the second big project initiative coming out of the ERGA consortium, where we're responding to the Horizon Europe call for European participation in global biodiversity genomics endeavors. And we took the bold leap of working together with the barcoding community and joining forces to create what we've called the project Biodiversity Genomics Europe, where we're really trying to work together to scale up and build this value chain of workflows that are required for scaling up capacity, production, and application, both in terms of producing reference genomes, but also in libraries of barcodes. And after a lot of work last year, we submitted the proposal, which was accepted. And now we start, in fact, this month, this joint proposal with a duration of 3.5 years. Next slide, please. So just to give you an overview of the kind of reach of this project, the Biodiversity Genomics Europe project, within the project consortium, we have 30 partner institutes from 20 different European countries, as well as 3 international partners. So this is sort of part of the remit of this project, was to be the European nodes of global efforts. So we partnered with the International Barcode of Life, the Earth Biogenome Project, and the Vertebrate Genomes Project, where beyond those that are directly involved in the BGE project, the ERGA network spans some 37 countries with 800 members, and the BioSCAN Europe network is already connected to over 100 different institutions from 29 countries across Europe. So together you can see that these two networks, if you like, bring people interested, researchers interested in biodiversity genomics together from across the whole of Europe. Next slide, please. So Claudio has already kind of summarized the main goals of the ERGA initiative, being the European node of the Earth Biogenome Project. And from the BioSCAN perspective, you can kind of see the mirroring effect where BioSCAN Europe is the European node of the International Barcode of Life project, which is a hub for DNA-based biodiversity monitoring, really connecting and enhancing all of the already existing and being developed national barcoding infrastructures. And we're not going to go into detail right now because you'll hear from some of the work package leaders with more details on that. But just as a brief overview, next slide. One of the main reasons for coming together from the reference genome community and the barcoding community is realizing the potential for improving the alignments, particularly at the beginning of the workflow. So with respect to species sampling, avoiding duplicating our sampling efforts on both of these networks, but also trying to develop protocols that are going to work for both getting good samples for producing reference genomes and for building libraries of barcodes. And clearly, with the species sampling tasks, there's a lot to be done in terms of streamlining the ethical and legal frameworks where we should be working together already. Further down the line, in terms of metadata processing, there's a lot of synergies where we need to improve the harmonization of metadata collection procedures and standards, and also standardize how these are processed all the way from the collection steps to the final deposition in public repositories. And this obviously also involves improved data integration to enhance the interoperability so that all of these resources that we're trying to produce are actually usable by researchers across different disciplines. Next slide, please. So this is a rather complicated slide, but the take-home message is that we have a workflow for producing the catalogs of barcode libraries and a workflow for producing the reference genomes. And in many senses, the steps along each of these workflows mirror each other. So from what we call the field to lab pillar, which is the sample collection, to the sequencing, to the data, to applications, and finally making use of these data in biodiversity applications. Next. And so for the purposes of today's webinar, I think it's fairly obvious that these work packages that are most relevant for the national focal points are the field to lab pillar. So the sampling on both the barcoding side and the reference genome side, but also on the left there, you'll see the work package 14, which is dedicated to policy and ethics. Next slide, please. So just very, very briefly, work package 4 is the barcode sampling to establish the supply chain for DNA barcoding to support this reference library construction and eventually applications in biomonitoring. Then on the genome sampling side, this is really aimed to deliver quality biological material for important species. So as Claudia mentioned, some of those target groups of organisms that have been identified by the European Union as a priority. And you will hear from both the NTNU, Norwegian University of Science and Technology, and the Museum Koenig with a few more details of the practicalities of the tasks involved in each of these main work packages. Next. Then realizing the fact that in Europe today, we don't have a very harmonized and streamlined or even coherent idea of the policy and ethics that we need to make this work. Work package 14 is really aiming to guide the policy direction, best practices, codes of conduct, etc., relevant to genomic research, kind of using the expertise or using the experience of the barcoding community and the genomes community to develop this policy framework. And obviously, this is working closely together with work package 4 and work package 5 to ensure compliance and coherence with existing policies, as well as developing a more solid framework. Next. So this is my final slide and a really sort of cartoon version of what it is that we're trying to achieve here, with the genomes and the barcoding networks or communities coming together to produce this first kind of stab at reference genomes for eukaryotic species across Europe and reference barcodes. And here focusing obviously today on this sampling coming from community sampling, what we call biodiversity hotspot sampling, but also the museum sampling for both genomes and barcodes. So hopefully that's an overview of the kind of main aims of the Biodiversity Genomics Europe project, which the work package 4 and work package 5 coordinators will be able to give a few more details on the implementations and the tasks involved. Thank you.
Thank you, Rob. That was a great description and definition of what Biodiversity Genomics Europe is. So next up, we have a presentation by Dr. Rebecca Uman, who is from the University of Oslo. And she is one of a number of people involved, heavily involved in the work package 5. And we'll discuss today the ERGA species sampling strategy proposed for the BGE project. So I will pass it over to you, Rebecca, and you have about 10 minutes.
Great, thanks, Anne. Yeah, so today I'll just give a brief overview of 2 of the main pathways for species sampling within the ERGA half of the BGE project. And those 2 pathways are for critical biodiversity sampling, and these are species that are nominated by the ERGA community from countries across Europe. And the second pathway is biodiversity hotspot sampling. And in this case, it's the locations that are selected, and then secondarily, a group of species are sampled from those hotspot locations. Once species are selected, they will automatically or immediately be tracked using publicly available software. They will be collected, they will be sequenced, they will be biobanked in terms of tissue and DNA vouchers, and there will also be specimen vouchers held at biobanks and photographic vouchers as well. And throughout this process, ERGA is using appropriate infrastructure to ensure robust management of all of the metadata. Ultimately, the genome sequences will be available. Next. So throughout this process, for both pathways, everyone will adhere to ERGA's Code of Conduct for sampling, including all ethical, legal, and social issues. The specific methods of sampling will necessarily be taxon dependent. Of course, it's slightly different for algae, for plants, and for vertebrates. But if possible, specimens will be collected and placed into liquid nitrogen. As adequate amount of tissue will be sampled for biobanking, as well as documenting the specimen via image, confirming the species identity via barcoding wherever possible, so that we can connect the barcodes to the reference genomes. And as well, there will be specifically DNA and tissue vouchering in biobanks as well. So the first major stream, of which there will be approximately 150 samples or species collected from across Europe. Samples in this critical biodiversity category, they're suggested by the community. From these 1,500 nominations we received, we will select about 150 to move ahead. And part of this species selection process will seek to balance representation of European countries in terms of the number of species sequenced from each country. And so the key takeaway here is that all European countries will be involved sooner or later, and most likely sooner. These species come, thank you, Anne, come from a broad range of taxonomic groups, the details of which are not so important today. Next. But one thing that is relevant is the main motivations and envision future applications of these reference genomes. So from these community samples, the primary reason for nomination is for conservation reasons, also to do with climate change impacts. Some are motivated by economic interest, some because they are pollinators, keystone species, or because they could be potentially used for pharmaceuticals. And these are, of course, not mutually exclusive categories. Next. One thing that can be of interest is that when nominators were asked how difficult it would be to obtain sampling permits such as Nagoya to allow the use of samples for reference genome generation, most felt that this would be that there would be either be no permits required, that there would be it would be a very easy and quick process with minimum delay, straightforward, weeks or up to a month, And very few envision this as being a challenging process, only about 5%. So it will be interesting to see how this aligns with reality in this large experiment we are undertaking. Thanks. The next category is biodiversity hotspots. Can move ahead there. So 3 hotspots have been selected so far from Spain, Greece, and Slovenia. And so there will be a greater number of samples sent from each of these countries to the appropriate sequencing centers across Europe. In addition, next slide. In addition to those 3 hotspots selected, there will be open calls for what we are calling BioBlitz events across other European biodiversity hotspots. And this is partly a citizen science engagement activity. So there will be 2 calls, one in late 2022 and one in 2023. Next. So of the species that have been selected from the 3 initial hotspots, the vast majority are endemic to Europe or European waters, or even endemic to the country that has nominated them. Next. The main motivations here are perhaps interestingly slightly different. It's because these species are of evolutionary interest in ongoing research groups. Of course, more are also of interest for conservation reasons because they are endemic or endangered or invasive. So there's a variety of reasons. And again, these categories may not necessarily be exclusive to one another. Next. Do you have or will you have the Nagoya permits for your species of interest? These are just from these 3 hotspots, remember. Interestingly, very few have them already. Most seem to think that they are not needed if the sequencing occurs in the same country where the species is from, which can be true, of course, but this will not be the case for the vast majority of species. So in fact, most will first need to know which sequencing facility their species will be sequenced at in order to apply for the Nagoya permit. And so this is something that we will have to undertake soon, as the sequencing centers are currently deciding on the allocation of species based on their sequencing expertise. Next. And most, nearly all of these species will be coming in over the next year, with about a third up until the end of 2022 and half in the first half of next year. So these are going to be coming across many of your desks in very short time. Thank you.
Thank you so much, Rebecca, for giving that overview of what ERGA has in mind for the BGE project samples and some of the challenges that we face as part of this project. That was a great overview and leads directly into our next panelist, who is Torbjørn Ekrem from NTNU. And Torbjørn is taking on the leadership of Work Package 4. And today he's going to give an overview of the sampling strategy for the BGE project, but coming from the BioSCAN side. So I'll pass it over to you, Torbjørn, and you have about 10 minutes as well.
Well, this is a slide we can quickly pass over. I'm the Work Package 4 lead. Here is a small table to show you how many institutions are involved in this particular work package. And they are listed here with their acronyms, as you can see, and also you can see the objective of this work package in the BGE project, to establish the sample supply chain for DNA barcoding to support reference library conduction and biomonitoring. So it involves quite a few different sampling types. So that's why I would like to go through very briefly some of the key points in each of these 5 tasks that we have before us in Work Package 4. The gap analysis, the metadata standards and biobanking, museum collection sampling, terrestrial field sampling, and aquatic field sampling. So next, please. Gap analysis, we have the Zoologische Staatssammlung München, SNSB, that is responsible for this task, but there are many collaborators in it as well. The whole idea is to provide a priority list of missing or underrepresented species in relevant public DNA sequence databases. So looking into the Barcode of Life data systems, BOLD, and also into GenBank, and potentially also UNITE as a fungal reference database, and see what's missing there compared to which species are supposed to live in the countries that we define as Europe. We will have a special focus on terrestrial insect pollinators and freshwater invertebrates, aquatic plants and fish, and marine fish and invertebrates used in European biomonitoring. And to some of you, this might be, a bit vague, but we know that the Europe scale biomonitoring for freshwater, for instance, is governed by the Water Framework Directive, and different countries have different priority species in there. So it's possible actually to find species lists on the European level of which ones to prioritize. And then whatever is missing, when we find out what's missing, we can identify the sample localities via the species distribution modeling, or through the databases of museum collections that are collaborating in the big project DISCO. Next, please. So within this task, we have already developed a draft tool to identify the gaps and trace the progress of selecting specimens, so that not multiple of our 13, 14 partners are working on the same species at the same time. It will be important for us. And some initial gap analysis of major insect pollinators is already done even before project startup. Next, please. So the metadata standards and biobanking task is governed by Museum König in Bonn, and will define standards on the collection of specimens and sample metadata that leading to these data tables that we use by all the project partners. So it's important, as it is for ERGA, to use the same type of input data sheets when we come to registering whatever we're going to barcode in the common databases. And this particular task will also coordinate the biobanking of collected and processed samples. So the Darwin core-based tables that are BOLD compatible are already ready with the metadata that we think are useful. And this particular data sheet includes then checkpoints for permits, both with regard to legal and ethical regulations, but without specifying specifically what permit has been obtained. For the museum collection sampling, we are aiming for about 33,000 expertly identified specimens from 11,000 species, and provide tissues then to the sequencing centers to fill the gaps in the barcode reference library. Depending on how many specimens we have included per species, we can also look closer into interspecific genetic diversity and maybe find some priority taxa also relevant for the ERGA stream in the Biodiversity Genomics Europe project. This will, of course, the task here will be in close collaboration with Task 4.4, which is terrestrial field sampling, and Task 4.5, which includes the aquatic field sampling, so that we don't aim for the same species, the same localities, and whatever we cannot collect freshly out in the field, which is very difficult now for many of the rare species, we can go into museum collections and do genome skimming of those specimens and not just standard PCR-based Sanger sequencing in order to retrieve the barcode fragments of these genomes. Next, please. So we here have a dynamic progress and reservation list developed, and that will prioritize this fresh material, use the museum collections the way we can, and genome skim these old specimens. Please. For the terrestrial field sampling that's governed by CBO in Portugal, we are aiming to sample terrestrial habitats to fill the gaps in the reference libraries, but also to develop metabarcoding biomonitoring workflows. So we will be here a focus on pollinators, about 6,000 specimens and species of management significance. And we will develop some protocols and pipelines for comparable community sampling, where the biomonitoring focus is on pollinator communities, ecological restoration sites, intensification, and climate change. So this is what we had in our applications, but through recent meetings, we see that there might be it might be a good idea to reduce the number of different focus communities so that we can increase the number of samples per type of community. The aquatic field sampling that I'm leading, this task aims to sample about 1,300 species, 3,900 individual specimens of these, and also coordinate and perform some sampling of environmental DNA from 55 sites to assess marine invasive species. And here we aim to do 5 ports throughout Europe, preferably in the different oceans that surrounds Europe, and do those quite extensively with project partners, while 50 harbors will be laid out as citizen science projects where citizens and NGOs can participate in contributing samples through to the BGE project, sampled in their home vicinities. So all in all, it will be about 525 samplings events then across Europe. Next, please. So just to show you a little bit of how we envision the museum sampling workflow, starting on top here with a gap analysis being performed compared to the master taxon list, where we can go further down, tag this with a museum label, and design a museum that will sample this particular species, select specimen, image them, database them, and tissue sample them. Database them according to the metadata standards here in task 4.2. And these metadata standards contain this confirmation of ethical and legal documentation. Then all of this and the tissue sample is sent to a sequencing lab. And it's important to understand that this shipment is accompanied by material transfer agreement for the biological material that is acceptable to transfer this across borders. And here also, the restrictions and permits and Nagoya agreements will come into play. This was developed by Ben Price at the NHM, who's leading the task on museum sampling. And basically, it's a very similar scenario that we have for the field sampling, with one of the differences being that we need to have the permits for collection and export ready before we actually sample the species that we're targeting in the field. Please. So a quick summary of the sampling strategy for the BioSCAN stream. We have a really large variety of samples. We have fresh specimens from the field, old specimens from museum with different, collection dates, either before or after the Nagoya. We have community samples, aquatic, terrestrial. It could be eDNA samples and so forth. So we need to consider all of these different sample types when acquiring permits and showing our intentions to send these samples either as specimens, tissues, extracts, or DNA libraries across borders. We have a very similar basic workflow for specimen-based sampling. So the workflow will be similar if you collect it in the field or if you collect the sample in a museum. Very many of the European large museum collections have not been digitized before, so we would need to digitize the label information anyways when including them in the project. And we also have a very similar basic workflow for community sample. But the community samples are quite different from the specimen samples. So in terms of metadata, so we need to separate them relatively clearly when we develop the metadata sheets, and also when we specify what's included in the shipments that are accompanied by material transfer agreements. And an example, and the draft material transfer agreement is already available as an attachment to the consortium agreement of the Biodiversity Genomics Europe project. So all partners in the project are already agreed on that this is something that we need to have when we send our samples around. Thank you very much.
Thank you so much, Torbjörn. That was great. And thank you for keeping on time as well. So I guess it's over to me at this point. My name is Anne McCartney. I chair the ERGA's Ethics, Legal and Social Issues Committee. And today I'm just going to give an overview of what was discussed today, and then we'll have a bit of a polling session, an interactive polling session with the NFPs to get some feedback from what your opinions on ERGA, the BGE, and BioSCAN sampling strategies that we've heard today. So I just want to take us back and simplify the process of what we're talking about here, particularly for the European Reference Genome Atlas. And so this is our life cycle, if you wish, for our projects. Each project starts with step one being permitting. We are thinking about, so obviously every sample, every species that we sample, no matter what country it is in or who collects it, we want to ensure that we are collecting and maintaining any permits that are needed or necessary or required. The next step in the process then is the sample collection and metadata collection. So each one of our species that has a sample associated to it will also have a very comprehensive metadata collected with it that will sit alongside each species to contextualize that information, giving it rightful attribution to the sample provider. And also, we didn't go into the details of the structure of what's within that manifest, but it covers things like country of origin, place of origin, geographic coordinates. It covers permitting obligations, biobank IDs, voucher IDs, indigenous rights and interests through the traditional knowledge and biocultural labels and notices. And so it's very robust. It has, I believe, 65 mandatory fields and over 80 fields of comprehensive metadata that contextualize each of our reference genomes. And so that metadata travels from the species through to the public archives that the reference genome ends up landing in, which in our case for ERGA is the European Nucleotide Archive. So after we collect our samples and our metadata, the next step obviously is to ensure that each one of these samples has an associated biobank or cryopreserved specimen and an associated voucher in a permanent collection in a museum. These biobank specimens and vouchers will have an ID, and as I mentioned before, that ID will be present when we collect the metadata. The next step being the samples then are sent to the sequencing center, or the DNA is sent to the sequencing center. It could be the sample itself being sent or the DNA being sent, depending on the project that we have in question. But here, DNA will be extracted, libraries will be prepared, and then the sequencing will be taking place. And we are ensuring that all of our reference genomes are in alignment with the Earth Biogenome Project's quality metrics for what a reference genome, what is a high quality reference genome. So we are meeting global standards for all of our reference genomes. Next, the next step is how we are sharing and managing the sharing of our data. So all ERGA reference genomes will go into the public archive. So this is Firstly, the metadata will go into biosamples, which is an EBI platform for metadata, and that each metadata entry, which is a biosample ID, will be associated with the genomics data being uploaded to the European Nucleotide Archive, which will get an ERGA bioproject ID. And so these IDs are very important because they are a way to associate a permanent identifier to the genomics information and to associate that back to the metadata information, which has important information to do with country of origin, for instance, but also that facilitates these permanent IDs being cited as part of data availability statements in peer-reviewed publications. And so could be a way to track the benefits of reference genomes to, let's say, a country that is participating. So after we share our data openly, we create our reference genome, and that's also openly shared with the community through the European Reference Genome Atlas. We then have over 800 biodiversity researchers that are willing and ready to take on and tackle the downstream analyses that can be associated with all of our reference genomes. And these can span a myriad of biological questions, from evolutionary questions to genetic diversity questions. And it's really to start capitalizing on the utilization of reference genomes as a tool for thinking about how to monitor and protect species, but also for conservation management as well. So that is one of the primary goals of how we are thinking about how to streamline our downstream analyses, and then hopefully all of our reference genomes will be associated with a peer-reviewed publication, and that could take many, many forms. So as the chair of the Ethics, Legal and Social Issues Committee, what we are really considering is how do we ensure that we are ethically and legally complying with all of our obligations across Europe, across all of our obligations with all of the different European countries that are participating across the data lifecycle, right from the permitting step, right through to how we are communicating our research after the fact. And so this is what our committee is really charged with. And this is has further complexity when you think about the stakeholders that are involved in each step, right? So you have a project lead that perhaps takes on the role of obtaining the necessary permitting. Usually we would ideally like this person to come from the country where the species is being collected. The next, we have the person who actually goes out and collects the sample and the associated metadata. That can be a very different person that actually has submitted the permit for that species to be collected. Third is who collects the metadata. That could be a person very different than actually who collected the metadata. Then the sample is stored, obviously, in a museum and biobank, as I mentioned. Then the sample is shipped where the DNA is extracted and sequenced, and this is probably done by a different biodiversity researcher, perhaps in a different country where the sequencing center is positioned. And then the data is uploaded completely publicly to the European Nucleotide Archive and is subject to their data sharing policies that are in place. The genome assembly is usually done by a bioinformatician on their cloud computing service, and their data analyses then could be carried out by a different biodiversity researcher. And then we're hoping that each person that contributed to each of our species projects would get fair and rightful attribution in a peer-reviewed publication. And so you can see that throughout the life cycle of each project, it's very complex. It includes very different countries and a lot of different individuals participating in each of these steps. So whilst ERGA has the grand and wonderful mission to be as inclusive as possible, ensuring that every single European country has the opportunity to participate through building an accessible infrastructure, that comes with challenges of ensuring that we are complying with any ethical and legal obligations that we have to ensure that we are doing this appropriately. And so our committee works on the interface of kind of science policy with interactions with CTAF, who Rob mentioned is a leading work package 14, and also has a lot of members from the DSI network as well. Thinking thoroughly about how we can streamline our ethical and legal obligations, particularly in the context of Nagoya permitting across countries, and how we are complying with any EU due diligence obligations in this space. So I do want to close with a poll to get some feedback and input from the national focal points. So if you are a national focal point, this is a completely anonymous poll. We don't know who is providing us feedback in real time, but I'll just invite you to click on the link. I can place it in the chat once more. Just give me one moment. Oh, Rob just placed it in, that's great. And I'll just place it in one more. So if you just go to that link, and I will stop sharing my screen for a moment, and I will reshare in 1 second. Now, if we have everybody just logging in, the first question is what country, so for the national focal points, what country are you representing today? So if you can just drop a pin to give us an indication of who we have in the room and what national focal points are being represented, that will really help us in understanding what type of feedback we are getting. So I'll just pause here for a few moments and let more pins drop on our screen. And this platform is super intuitive. You just click on the link and it just opens up and you can participate. Again, completely anonymous, and we're not tracking who is participating here. So I see we have some people from the UK. We have some Spanish participants. Okay, I'll just move to the next question because we're in the interest of time. Is this your first time hearing about the European Reference Genome Atlas? So if you could click yes or no to give us an idea of, have you engaged with the project before? So wonderful that a lot of folks in the room have actually heard about the project before, which means that we are building awareness and hopefully starting we are communicating well. My next question is, would you be open to presenting your access and benefit sharing procedures to ERGA if a webinar series was organized? So is that we have shared a lot of information with you about what our ERGA has in mind today. And we would love for this to be a flow of communication that we could also learn about your procedures and how best we can comply with ABS obligations. And so just getting an idea of if this is an interest for you as a national focal point. So I see half are interested, which is wonderful. And 20% said yes, but maybe they would like to send a delegate in their place, which is great. So if we did host a webinar, how often do you think that would be a good time frame for that webinar to be hosted? So I'm seeing about every 6 months, we could potentially compress a number of focal points speaking at during one webinar, which would be ideal and a good utilization of your time. Okay, so my next question, if in one word, because this is a word cloud function, after hearing the discussion today, what are your priorities about ERGA? So when you heard the speakers present their information today, if you had one word, what in your mind, what would be a priority when you think about ERGA? So the generation of publicly available reference genomes. Consistency, that's nice. Comparability. Just in the interest of time, because I know we have to capacity, which is very important. I know a lot of C's coming up here, the 3 C's. Oh, we've got a lot coming in now. So taxa, data, ecosystems, very good. Included. Okay, that gives us a lot of information to work with. And our final polling question today is, after hearing the discussion today, in one word, what are your concerns about ERGA? So if you had a concern or a challenge that you foresee with generating these reference genomes, what would that be? So I see free data access coming up, which absolutely. Capacity, yes. Protocols, yep. Openness, participation, very important. Perfect. I just want to be respectful of people's time, but I also want to give people an opportunity at this point if they had any questions that they would like to share. Just looking at the Q&A, I don't see any questions that have been proposed in the Q&A function. But at this time, if you'd like to verbalize a question to any of our panelists, please raise your hand and we'll be happy to answer any questions about what you've heard today, or you can alternatively place it into the Q&A function. And we will pose those questions to the panelists directly. So I'll just give a few minutes. I've got one question. Could we hear some details on the BioBlitz? So for work package 5, I'm going to pose that question to you. Perhaps maybe Astrid or Rebecca, could you speak a bit more about what ERGA has, the BGE project has in mind for these citizen science run BioBlitz events?
Astrid, did you want to go ahead here?
Yeah, so what we have in mind is dedicated excursions to biodiversity hotspots, and if possible, have feedback from citizen science. Maybe Torbjörn can say more to citizen science. There's also involvement in BioSCAN, but so we have dedicated sampling campaigns in what we consider to be European biodiversity hotspots. Not all of them have been defined yet. So Rebecca presented 3 places we're going to, and the idea here is to sample as comprehensively as possible, always with a methodology that allows genome sequencing. Probably sample more than we will be able to sequence with current funding, but with the opportunity to sample as much as an ecosystem we could possibly sequence at some point. I don't know, Torbjörn, do you want to comment on citizen science a bit more?
Yeah, maybe we have a special task in the coordination structure where that is focused on citizen science, where the idea is to develop a citizen science web portal where the citizens, can track their own samples, where they submit it and what it has become, and when the sequences are back and what it matches up to and how it is used. So that is on the block to develop now over the next few months.
Thank you, Torbjørn. I have another question from Louis Fresta. Is the number of partners in the ERGA project expected to grow over time as more institutes, researchers gain interest? So maybe one of the chairs, either Claudio or Rob, I'll field that one to you.
Yes, I can give a short answer to that, and I'll hand it over to Rob for some more comments. Yes, I think so. If we're talking about the ERGA in general, we're not talking about the BG project, of course, although the BG project probably will have some follow-ups, but as far as the ERGA concerned, yes, we hope so. Also, considering that during the BG project, probably we'll try, one of the aim also of the BG project is to give a more formal structure to ERGA, which is now mainly an initiative comprised by, say, people, but we don't have yet say a legal or formal structure, agreements or such. So in that case, also we could that could be a way also of engaging more institutions and more people on the ERGA. Yeah, but the aim is to increase the network, yeah, of people and institutions. Yeah.
Rob, did you have any follow-ups?
Just simply to add that the kind of nucleus of researchers that got ERGA up and running and started is really centered around the kind of genomics, so the production of the data, the assembly of the reference genomes, and their annotation. And as we've seen, this is a much broader workflow from the beginning with sample collection all the way to the end with data analysis. And so the ERGA kind of consortium hopes to attract more researchers and more institutions from across the whole workflow, not just with this current nucleus focused very much on genome assembly.
Yeah, absolutely. So thank you so much. And I want to close this out by thanking again all of our panelists for their wonderful presentations today. I'd like to also thank all of the national focal points and ERGA members who have actually spent the last hour with us. We really, really appreciate that. As I mentioned before, we're going to follow up with the recording and the slide deck with you, and also hopefully start a communication of how we can build out, keep the flow of communication going between ERGA, the BGE, BioSCAN, and the national focal points to try and streamline and build capacity for what our ABS obligations are when we think about reference genomes and barcoding at scale. So again, thank you very much, and I will be in contact through email.
Thanks for organizing, Anne.
Thank you. Thank you.
Thank
Bye.
you.